Blog Talk Radio.
Welcome to today's episode of mPatient Myeloma Radio, a show that connects patients with myeloma researchers. We started this series to help you learn more about the latest research in myeloma so you can make your very best treatment decisions. We strongly encourage you to consider clinical trials as you think about treatment options. When we started this series, less than 5% of patients participated, and if we doubled that number, we could accelerate the pace that researchers can find cures. Now, if you'd like to receive a weekly email about past and upcoming interviews, you can subscribe to our mPatient Minute newsletter on the homepage or follow us there on Facebook or Twitter. And we encourage you to share these interviews with your myeloma friends. We also have a new site called myelomacrowd.org. That's the first all-inclusive and comprehensive site for myeloma. So please take some time to explore the links. There are listings for all relevant and very credible myeloma news sources, lists of who to follow on social media, growing online myeloma specialist directory, current articles on relevant myeloma topics, and links to all the good being done by the various organizations and foundations in myeloma. You can also contribute what you're learning by clicking on the Become a Contributor link. Now, we are very honored to have with us today Dr. Walter Michael Kuehl of the National Cancer Institute. So welcome.
Thank you.
Let me give a little introduction for you before we get started. Dr. Kuehl is the Chief of Molecular Pathogenesis of the Myeloma Section in the Center for Cancer Research at the National Cancer Institute. Dr. Kuehl obtained a Harvard degree in biomedical sciences and his medical degree at Harvard Medical School. He performed his residency in internal medicine at Case Western Reserve and completed postdoctoral fellowships at the NIH and Albert Einstein College of Medicine. He joined the Department of Microbiology at the University of Virginia Medical School and attained the rank of professor before joining the NCI. As head of this group and senior investigator, his research has laid foundational work to identify myeloma not as a single disease, but a disease with varying characteristics, or what you might think of as myeloma subtypes. With this, he also identified specific translocations and genes that drive the growth of myeloma, identified that MGUS almost always precedes myeloma, and identified specific pathways that are uniquely affected by the various types of myeloma. He's written over 150 publications, is the recipient of MMRF grants, and received the Lifetime Achievement Waldenström's Award from the International Myeloma Foundation. So, Dr. Kuehl, thank you so much for your detailed work in the biology of myeloma.
Well, it's a pleasure to be here. And do you want me to say something now?
Yes. Maybe you can kind of explain what the Center for Cancer Research does, and give us a short overview of your focus. Okay.
Well, the NCI, the National Cancer Institute, has gotten reorganized numerous times over the 25-year period that I've been here. And most recently, about 5 or 6 years ago, I don't remember the exact date, they decided to reorganize in a new way, and so it broke down into two main divisions. Or there's a Center for Cancer Research, and then there's the Division of Cancer Epidemiology and Genetics, which is focused on who gets the disease, what are the population characteristics, and what are genetic factors that might predispose to the disease, and so on. And that's a very small entity. The Center for Cancer Research is the much larger entity, and that's divided into a clinical and a basic science in basic science part. So the clinical would do translational research. They would do some clinical trials, and it would be focused on that. The basic science would focus on the fundamental mechanisms, not only in cancer cells, but in normal cells and even in lower organisms, so that we could better understand the biology of what makes cells grow and survive and so on. In any case, so that's enough about that. And so I'm in the clinical part of the Center for Cancer Research, and I'm in the genetics branch, which is focused on cancer genetics. And I want to start off by just saying that that the way I started to work on myeloma it turns out to be somewhat interesting. A number of years ago, I was one of the fellows in my lab, Leif Bergsago, who recently was on your show. We were sitting at lunch one day, and he says to me, "Why don't you study myeloma?" Leif's father was like one of the absolute giants in the first designing some of the first treatment for myeloma. And I said, the problem is it's hard to get adequate material. I said, if we could get cell lines, then I think I would really want it. I would definitely start to study human myeloma. I was studying mouse tumors mostly at that point. And he, being a very inventive person, he went to the computer, which I hardly even knew how to turn on a computer then. And he came up with 50 different myeloma cell lines that had been isolated but no one studied. So as soon as he said that, I said, all right, I'm going to stop everything in my lab and we're going to start working on myeloma, which is what we did. And we sat down and we said, what's important in what might be important in trying to understand it. And we decided based on other kinds of tumors, there were two things that we wanted to focus on. One was translocations, chromosomal translocations, that involved immunoglobulin loci, and two was MYC, because both of those had been important in mouse plasmacytomas and other kinds of tumors. And so that's what my work has focused on, is using that as a springboard, using the cell lines initially, but always realizing the cell lines do not represent exactly what you see in the tumor. They're actually an advanced form of myeloma. They're sort of the terminal stages of myeloma. Those are the tumors that go to extramedullary sites or plasma cell leukemia, and those are what give you cell lines. So they're very advanced. But most all of the early events, including the primary translocations and abnormalities of MYC, which is a later event usually, are immortalized in those cell lines. So it's kind of like an archaeological dig, essentially, to see what's there, and then take that information and go to look at the tumors at different stages, including pre-malignant MGUS even. And so that's what we've done. And so we started everything in the cell lines, and interestingly, for a long time there were people who were, even though cell lines had been used for many years by many people, there were skeptics out there who said, they always would say, oh, well, the cell lines don't mean anything. I mean, you have to study the real tumor. But the reality was that what we found in the cell lines, we've never found any major pathogenic change in a tumor that we didn't verify in a cell line that we didn't verify in a tumor. Now, that being said, cell lines do not represent the full spectrum of the disease. And there are certain types of myeloma, like hyperdiploid, which we can talk more about that later, seem to be underrepresented among the cell lines. And the ones the primary translocations, which we started working on initially, are overrepresented. And I can explain reasons for why that is. But the bottom line is that the cell lines are not a perfect mimic of the tumors, certainly at the various earliest stages of the disease. But they give us a window into trying to understand how we got there. And so, that being said, our initial focus was then to focus on IGH translocations. So these are on chromosome 14. And the IGH locus has very strong regulatory elements that increase the readout of genes in the immunoglobulin locus. And what a translocation does is it moves other genes, oncogenes in this case, closer to the immunoglobulin locus on chromosome 14, and so that those regulatory elements which cause high levels of RNA expression now are imposed on other genes that are brought in. And so a translocation, of course, is a rearrangement between two different chromosomes which bring a particular gene region on a different chromosome into the immunoglobulin domain so that it can be regulated by the immunoglobulin regulatory elements, and then you get very high levels of expression that don't change during the cell cycle, so they're relatively constant and very high levels. And in the process of doing that, what we determined was that we found that as it turned out, about 40% of myeloma tumors have a primary IGH translocation. Now, what do I mean by primary? Primary, it doesn't necessarily mean it's the initiating event, although they might well be. But what it means is that they're translocations that are commonly found both in myeloma and in pre-malignant MGUS. And so we call those primary, even though we can't prove that they're the first event, but they're the first event that has been identified by anyone. So we found three. So there were 7 translocations, which are in 3 groups. There's a cyclin D group, the 3 cyclin D genes, D1, D2, D3, and then there's a MAF group, and there's 3 MAF genes. There's MAF, there's MAF-B, and there's MAF-A. And then finally there's a group called the MMSET FGFR3 group, and so that's the other group. The cyclin D1 is by far the most common, and the MMSET FGFR3 is second most common, and then MAF and MAF-B are less common, and the others are relatively infrequent in the range of 1 or 2% of tumors. So what are the characters? So one of the other things is that we got interested also in other kinds of rearrangements, and there are what we call secondary rearrangements. Well, all that means is that from our point of view, is they number one, they tend to be present in some myeloma tumors, but not in the pre-malignant MGUS. Number 2, that sometimes if you're lucky enough to have samples from a patient that you can observe over a period of time, that you can show that those rearrangements are acquired with time. And one of the best examples of that is the MYC oncogene, which actually we have actually, and others have observed, that those rearrangements come up during the progression of the tumor. But they may also come up earlier, which, we can talk more about that. So at least in a nutshell, and the other thing that I should just point out is that primary translocations generally are very simple exchanges between 2 different chromosomes. They are thought to be mediated primarily by errors in normal mechanisms in B cells, in germinal center B cells, that undergo different or earlier cells possibly, that undergo 3 specific DNA modifications. So one is called VDJ recombination. That's what makes the antigen receptor part of the heavy chain or light chain in the immunoglobulin molecule. Another is somatic hypermutation, which is somatically hypermutating the antigen binding site in the antibody. That generally occurs mainly in the germinal center. And the third is switch recombination, IgH switch recombination, which is involved in changing from one isotype of immunoglobulin like IgM to make other isotypes like, for instance, IgG or IgA. And all of these 3 events all involve rearrangements or modification of DNA that create double-stranded breaks in the DNA, and that then predisposes to translocations. So most primary translocations appear to be mediated by one of those processes. Most of those are thought to occur mainly in the germinal center. So this is consistent with the idea that a myeloma is a tumor that is whose normal equivalent is a long-lived plasma cell that has undergone modifications by those 3 processes, and it happens mainly in a germinal center. So the thought is, that's why we think that is certainly a very early, if not the initiating event of the tumor.
And do you have any thoughts about what causes those primary?
Well, not really, but I think that there are errors that are rare. But what can happen is those 3 mechanisms can spill over into genes other than the immunoglobulin locus, so they're not perfect. And so 2 things happen. One is when you create a break, if something disrupts the rapid repair of it, or the normal process that occurs, it's hanging around, and then it can pick up some other piece of break in another piece of DNA. Secondly, those mechanisms can spill over and sometimes affect not just the immunoglobulin genes, but other genes. All of this is rare events. These are errors. So, in other words, most of the time things work out smoothly, but occasionally errors are made, and what we think is that the primary translocations are mediated by errors in these B cell specific mechanisms. By contrast, I should just add that secondary translocations occur beyond the germinal center, when you already probably have an MGUS tumor or beyond, and those mechanisms are not on there. And lo and behold, when you look at the rearrangements there, number one, they're very complicated. We don't fully understand that, but more importantly, they don't have the hallmarks that you would expect for these B cell specific DNA modification mechanisms. Does that make sense?
Yeah, it does. And is one worse than another? It's worse to have these as primary translocations, or it's worse to have them as secondary?
Well, I mean, I don't think worse would be the word I would use. What I would say is that the primary translocation is what gets you started. I mean, and they're found in MGUS. So if you have, and as you know, MGUS is a pretty benign disease, and in fact, I think even though it's a very common disease, and people think about treating it, I mean, at this point, people aren't treating MGUS except MGUS cases which have a high likelihood of progressing. Which isn't so easy, but I mean, that's something we could talk about, or you could talk about it another time. But I think what's important to realize is that if you were a 55-year-old person who had MGUS, your life expectancy is no different than if you didn't have MGUS. It's marginally different. It's hardly noticeably different. So MGUS generally is a very benign condition. So these translocations give you that first step, and then it's the secondary events that lead you on to myeloma. One of which is what we think is that second event that causes you into this. So I divide myeloma into three transitions. So the first transition is what gives you MGUS. So primary translocations or hyperdiploidy, which we can talk more about that. And then that's the first transition, to get from a normal cell or pre-MGUS cell to a pre-malignant MGUS cell. The second transition is to go from MGUS to myeloma. Now, unfortunately, that's a clinical definition, and so it's very hard to say if you had a patient with a tumor and there were no secondary manifestations like anemia or bone disease, etc., if you handed me those cells, or anyone those cells, they would be hard-pressed to say whether that cell was myeloma or MGUS. So it's a clinical definition, which means that unfortunately I can't say this cell is MGUS or this cell is myeloma most of the time. I mean, if it's an advanced myeloma, maybe I could. But in any case, what we know is that as you go from MGUS to myeloma, that myeloma tumors express about a threefold higher level of MYC on average than tumors that don't have that are MGUS. And similarly, what we find. We have shown that KRAS mutations are present in myeloma, but we have never found them in MGUS. So the thought is that the progression, that second transition from MGUS to myeloma, it could be mediated and requires the upregulation of MYC, so it makes more MYC RNA, and that KRAS mutations could also be involved in that transition. Although there's not a cause and effect that we can prove particularly for the KRAS. Now, there is an animal model for MYC which suggests that an MGUS-prone mouse, if you upregulate MYC in it, which I mean Leif, I'm sure, talked about when he was on your program, it's a model they developed. Those animals now get myeloma. Before they just got MGUS, but when you upregulate the MYC, they get myeloma. So and that's consistent with what we see in the human and the human MGUS and myeloma cells, namely that MYC is upregulated. Let me just mention the third transition. There's a lot that goes on in myeloma, many other molecular events that you can ask me about. But my third transition is the transition of going from a relatively quiescent state to a proliferative state. And proliferative meaning that the cells are dividing. Myeloma and MGUS basically are cells that mostly are just sitting there, and they're not in cell cycle, they're not replicating. Even though to get where they are, they have to have had a lot of cycles of replicating. But at a point in time, very few of the cells are in cell cycle and are synthesizing DNA or dividing. But late in the disease, you develop extramedullary myeloma or plasma cell leukemia, and typically that's associated with an increase in proliferation. And that is mediated by a variety of changes, one of which we've determined is disruption of the RB pathway beyond cyclin D dysregulation. And we can talk more about that. P53 mutations also tend to happen at that more at that later stage of disease. So in any case, I think of that as an important transition because it's going from a relatively non-proliferative state to a very aggressive state and more likely to create more problems for the patient.
Mm-hmm. And I've read it does. And I've read about growth factors, and maybe they have something to do with that. The FGFR3 and
yes. Well, okay, growth factors. I think one of the most important things to remember about normal plasma cells, but it's also true for MGUS and for myeloma, particularly at the earliest stages, they're absolutely dependent on the bone marrow microenvironment. And so all the cells that are in the bone marrow are cooperating with the tumor to sustain the tumor. The thought is that normally there's a niche in the bone marrow for plasma cells and certain other kinds of hematopoietic cells. But normal plasma cells are only about 0.5 to 1% of the cells in the bone marrow. An MGUS tumor can be up to 10%, and a myeloma can go 30, 40, 50, even 70 or 80%, at which time you start to see major problems with interfering with blood formation. Mm-hmm. And the thought is, but until very late in the disease, typically, the tumor is confined to the bone marrow. It's dependent on that niche, but the niche is expanded. It's much larger than it would be for a normal plasma cell. And we don't really understand how that happens, but the thought is that to some extent the tumor can become less dependent on the bone marrow, even though it still has some dependence, so it can't survive very well outside of the bone marrow. But there's also this idea, again, not fully proven, but there's certainly supportive evidence that the tumor reprograms the bone marrow and makes it more inviting and more effective to support the tumor. And in fact, while it does that, in some cases you see, even in MGUS, for example, you'll see an increase in normal plasma cells. Presumably, when it changes the niche, the tumor, it also can increase the number of normal plasma cells. Although ultimately, as the tumor progresses, it crowds out the normal plasma cells, which creates one of your problems with immunity in patients. So factors are very important. Now, you mentioned FGFR3. And so there's sort of been a controversy a little bit about, that was one of the first translocations that we identified, and it actually is an unusual translocation in that it goes into a switch region, an IGH switch region, and it divides the regulatory elements onto the two chromosome derivatives. So one of those regulatory elements dysregulates FGFR3, which is ectopically expressed, is very high. The other dysregulates mMSET, which is a chromatin modifying gene. And but it's clear that the mMSET is the more important of the two, in the sense that every cell line and tumor that has the 414 translocation, you always retain the chromosome that is dysregulated for mMSET. But in about 20% of tumors, you lose the chromosome that dysregulates FGFR3. So you might say, well, FGFR3 isn't important. I think that's wrong. I think it is important, but I think that it's important only in the initial stages of the disease, and that there are other things can take its place later on. A RAS mutation, for instance, can eliminate the need for the FGFR3 and other kinds of things. And there's definitely evidence, to support this idea, and I think most people would probably agree that the dysregulation of both genes is critical in the initiation of the disease. Does that make sense?
Yeah, it does. And just overall, a general comment, I think the discovery that there are these different mutations, different translocations, that you've subdivided them, is really spectacular and stunning. And I would think, from your perspective, what does it change in terms of your research so far?
Well, I mean, I think what it does is I think it really demonstrated to me that myeloma really is different than molecular diseases. So there's a type of myeloma that has a cyclin D translocation, and there's another one that has a MAF translocation. There's another one that has the MMSET FGFR3 translocation, and then there's another group that's called hyperdiploid. And so what is that? Well, we don't understand that one very well. But what it is it has extra copies of some combination of 8 odd-numbered chromosomes, and so that you have extra copies, so 3 copies instead of 2, typically. Sometimes you can have 4 copies instead of 2. And again, what does that mean? We don't really know what that means, but it's an absolute invariant finding. There is a class of tumors that have that, and we call those hyperdiploid. They tend to have a better prognosis. The thought is that they're possibly more stromal cell dependent. They're underrepresented in extramedullary tumor or plasma cell leukemia. They're underrepresented in cell lines, and so they're a little bit mysterious. But one of the things that one of the unifying theories that we came up with, and which we think is right, is that there's a virtually universal dysregulation of a cyclin D gene, either whether you have a translocation or you're hyperdiploid, or the occasional tumor that's not hyperdiploid and doesn't have a translocation. So most translocations are in the non-hyperdiploid tumors, most of the primary translocations, and the hyperdiploid tumors tend to not have primary translocations. But what's common is that in all tumors you dysregulate cyclin D1, D2, or D3.
And can you explain what those do and what they are?
Okay, cyclin D is critical in regulating the proliferation of cells, so that if you upregulate cyclin D, it can interact and activate the so-called retinoblastoma pathway. And so the idea is, for a long time, people thought that all you have to do is upregulate cyclin D by it could be by a direct translocation, MAF. The MAF translocation, by the way, MAF is a transcription factor for cyclin D too, so that's how that gets upregulated. And in the hyperdiploid, they ectopically make biallelic. That means both alleles, so not through a translocation or some obvious mutation in the gene itself, upregulate cyclin D1. And what this does, so this virtually all, like 95% plus of tumors have upregulated cyclin D1 or D2, and rarely cyclin D3. Even though normal plasma cells make cyclin D2 and D3, the cyclins that are dysregulated in myeloma are made at a higher level than in normal plasma cells. Now, upregulating cyclin D is not sufficient, though, to drive the cell to rapidly proliferate, because there are still checks on that. There are genes that can inhibit that interaction of cyclin D with I'm not going to get into detail here, but they basically interfere with its action. So the thought is that, as I said before, myeloma cells and MGUS cells are relatively non-proliferative. Okay, so the cyclin D doesn't seem to be pushing them into proliferation. On the other hand, I think what the upregulation of cyclin D does is it makes the cells more susceptible to external signals and internal signals that periodically stimulate it to go into proliferation. And so, even though most of the cells aren't proliferating, I think that inexorably there's a slow amount of proliferation due to cells that occasionally enter the cell cycle through mechanisms we don't really understand, but presumably through various extracellular factors that are stimulating the cells, and because they are making more of one of the cyclin D genes, they're more susceptible to entering into the replication pathway, making DNA and going on and dividing. Okay. When I talked about the transition 3, when a tumor becomes more proliferative, now at least in many cases, we know there's a second disrupting event in the RB pathway. That is the pathway that regulates proliferation, and that can happen by either a mutation, inactivation of RB itself, which is kind of the key molecule in this. Or there are other inhibitors that basically block the effective interaction of cyclin D to be able to activate the pathway. One of which is what we call INK4C or P18 is the one that there's actually different kinds of that for different sorts of cells, but in plasma cells, the P18 is really what's important. And so what we found is in proliferative tumors that about 25 to 30% of them have inactivated that gene by deleting it, completely deleting it from the cell. So basically we've developed the concept that the cyclin D dysregulation is key for an initial event that's common to MGUS and myeloma, but it doesn't cause a lot of proliferation, but probably enables the cell to proliferate more than a normal cell, and that at later stages you further disrupt the pathway. Now the tumor can become more proliferative. That's the basic idea. And,
so a question
now,
is.
Go ahead. Okay. Sorry.
I guess, no, I'm sorry to interrupt you. I'm wondering, now that you know the cyclin D is kind of the start but not the accelerator, and the RB pathway might be the accelerator, are there things that have been found that you could shut that down early, like at the cyclin D stage?
Well, people have tried to develop inhibitors. There are inhibitors against the molecules that cyclin D interacts with, which are called CDK or cyclin D kinases, and there's a CDK4 and CDK6. So people have developed inhibitors against those, but they're not specific for the tumor cell. In other words, they would affect all proliferating cells. It's kind of like chemotherapy. A lot of chemotherapy is non-specific, so it's targeted against proliferating cells, and as a result, there's significant toxicity associated with it. And as you probably know, it interferes with your ability to create hematopoietic cells, red blood cells and white blood cells. So there is, unfortunately, at this time, there is no really good drug that can inhibit the effect of cyclin D1 or D2. I mean, even though people are trying with the CDK inhibitors, they're trying to use them to treat, but unfortunately they're not highly specific, and there's significant side effects. Does that make sense?
Yeah, it does. And then the RB pathway, is there anything that you know of that stops that at that point?
No, there are no specific drugs that can. I mean, part of the problem here is as you know, there's oncogenes and there's tumor suppressor genes. Mm-hmm. So the RB itself is a tumor suppressor gene, so it's standing in the way of the cell going on to proliferate. So if you inactivate it by either homozygous deletion or mutation, it's very hard to replace a gene that's lost from the cell. And it's the same way with these other molecules, the P18 I talked about. Mm-hmm. Again, that's a tumor suppressor gene, and it's lost from the cell. You can't bring it back easily. So you basically need to find some other way to take advantage of the fact that cell has lost the RB or the P18, and the hope is you find that makes the cell more sensitive to some particular agent. That's the dream, but it's tougher than that. And there aren't the bottom line is there aren't good drugs that specifically target the RB pathway. Mm-hmm. And so often what's used is therapies that are the chemotherapy that's very toxic. It's somewhat nonspecific, but with transplants, with auto transplants, it's possible to overcome some of the side effects of that therapy. I mean, the blood forming cells being the most sensitive, and so that's what the transplant does. Is it restores blood cells that you've killed off with the therapy, and hopefully you've killed off most of the myeloma tumor cells.
Okay, and now I don't want to stop you from what you were talking about before. My apologies. If you can remember.
Oh no, I think the other thing I was just going to point out is that, so you asked me how having the different kinds of myeloma impacts research. Well, I think what's really important is that when you start looking at trials, for instance, that it's important to recognize that myeloma is multiple diseases, and so a given drug might work even if it's not directly targeted against one type of myeloma. It still might work better against one than another. And in other words, it might work better against the ones that have the MAF translocation than it does against the ones that have the cyclin D1 or vice versa. So I think what's important is to appreciate the fact that you're dealing here now with multiple diseases. And for instance, when you do clinical trials, if you looked at it's possible that if you look at the myeloma tumors that you haven't classified, that you won't identify something that might give you a positive effect. Let's say on the one that have MAF translocations, for example, so that's like 6 or 7% of tumors. So if that 5 or 6% is buried in there, along with the other 95% that don't have that, you won't see the effect. I mean, you'd have to have an incredibly large study, which doesn't usually happen. So I think it's very important to recognize that this is different diseases, and it's even more complicated than that in the sense that. So we developed a classification which we called TC, which was based on the translocations and cyclin D1 expression as RNA. But and that holds up for both MGUS and myeloma. So it's looking at the early events. But what's important is that there are later events that happen, and that's going to ultimately that becomes important too. So I mean, just as an example, about 40% of myeloma tumors have an N or a KRAS mutation. Now, if you had a therapy, and there isn't one, by the way, and there's a lot of work going on here now trying to develop one, but if you had a RAS mutation for any kind of particular myeloma tumor, and you had a specific drug that targeted RAS, then the most important thing would be to identify the tumors that had the RAS mutation. And and that goes on for all of the other kinds of things you see. So there are, one of the kinds of mutations that actually that we came upon at one point was NF-kappa B activating mutations. And so NF-kappa B is a transcription factor that's tightly regulated, and if you activate it, which in normal plasma cells can be activated by signals from the microenvironment and in tumor cells. But as the cells become more autonomous, it turns out that it's advantageous for the tumor to pick up mutations in regulators of NF-kappa B. And there are many regulators, but there are mutations in like 8 or 10 components of the NF-kappa B pathway, and those mutations cause the upregulation and activation of NF-kappa B transcription activity. And what that does is it affects genes, it changes the expression of genes that favor the survival of cells mostly. Mm-hmm. And so therefore the cells are less likely to die, and this is really key even in normal plasma cells, but it's done by the microenvironment. It's done by the bone marrow stromal cells that release factors that activate NF-kappa B through external signals. But as a tumor, and that's true in the MGUS and the myeloma tumors as well. But as they progress and particularly when they become very advanced, the NF-kappa B activation that's a result of mutations in the cell makes that cell not dependent on the signals from the microenvironment as much. So I don't know if that makes sense, but tumors in general strive to become more and more autonomous, mm-hmm. and that's as I said early on, they're completely dependent on the bone marrow microenvironment, and they only can be in MGUS there may be 5% of the cells or less in the bone marrow, and then as you go on, they take up more and more space in the bone marrow, so they have less dependence, and then ultimately they can move to extramedullary sites, meaning outside the bone marrow, or to plasma cell leukemia, and at that point they no longer can rely on the signals that they got from the bone marrow, and now they need to be able to provide their own signals. And so the NF-kappa B mutations that deregulate that pathway and cause increased activity of NF-kappa B are just one example of that. Mm-hmm. I think the loss of FGFR3 is similar in that a RAS mutation, for instance, might replace FGFR3. And so, and actually, the other thing about FGFR3 is it's a receptor. It's fibroblast growth factor receptor 3, and so it requires a ligand. It means something to stimulate a molecule to hit it and stimulate it, and there are certain cytokines then that so-called FGFs, fibroblast growth factors, that can stimulate it, and so that means that if the FGFR3 were critical for the survival of that tumor, it would then require those signals. So it gets around that by a RAS mutation or something else. So eventually FGFR3 becomes dispensable. Although I will also say another way that it becomes actually critical is that if you have a mutation in the receptor itself, that will activate the receptor. No longer needs signals from the outside, and so that happens too.
Mm-hmm. And in the NF-kappa B pathway and the RAS mutations and some of these other mutations, how are you testing, and the cyclin D, how are you identifying those? Are those through the flow cytometry test, the FISH test, the gene expression profiling? How are you
gene expression profiling? Mm-hmm. Number one, it's directed PCR and mutational analysis. It's sometimes through FISH, but I think more nowadays it's more through comparative genomic hybridization. I mean, a lot of this was discovered by copy number changes, and you could show like homozygous loss of both copies of a gene or one copy of a gene, and then mutational analysis. And I mean, now, as you know, I mean, everything is being sequenced left and right. Mm-hmm. Although it's interesting, I think that a lot of the most important things were discovered before whole genome sequencing came along, and so the whole genome or exome sequencing I think has uncovered and shown and demonstrated very clearly that there's a lot of heterogeneity within the tumor, and that some of that was not discernible by the earlier studies. But the key genes that are involved, many of them were found before whole genome sequencing, and not to say that there haven't been some new ones.
Well, from your cell lines, right? That's what
yes.
That's where you.
Yeah,
found.
I mean, almost all of the abnormalities that one sees you can find in one or another cell line, and what you're talking about mostly is the prevalence or frequency of things. So you might find that some particular thing, well, it's like the inactivation of P18. So cell lines are proliferative, and so about 30% of the cell lines have homozygously inactivated P18, mostly by homozygous deletion. In the tumors, it's only about 5%. But if you pull out the tumors that are proliferative, which you can do by looking at their what genes they make and so on, or well, there are other ways to do it. Now, all of a sudden, you see those homozygous deletions of P18 that are similar to what you see in the cell lines. Does that make sense?
Yeah, it does. Can I ask a question about a little bit more about testing? I know there's a new testing, I think it's from the binding site, called the heavy light chain test.
Right.
And since these are heavy chain translocations, is that test important? Could you explain if that test is relevant or what that test does?
No, I don't think it's related to translocations.
Okay.
I mean, I'm not that up on that test, but what that test is doing, I shouldn't talk too much about it. I should know more about it, but I'm not fully up on it. But I think basically the idea is that there was a light chain assay. There's a free light chain assay, okay, which is very sensitive, and the heavy chain, adding the heavy chain in, it just increases it increases your sensitivity, because now you're looking at a specific subgroup of the heavy chains. There are different kinds of heavy chains, and now if there's what are called different isotypes. So there's alpha heavy chains for IgA, there's mu heavy chains for IgM, there's gamma heavy chains for IgG, and there's like 4 different kinds of gamma, and there's delta chains for IgD, and there's epsilon chains for So what happens is if you look at all the IgG at the same time, for example, you're looking at 4 different kinds of IgG for the 4 different gamma chains. Okay, there's IgG 1, 2, 3, and 4. So by having being able to look at a single heavy chain, you can develop more sensitive assays, because now you've just narrowed the window of what you're looking at. Does that make sense?
Yeah, it does. And I know
it doesn't have anything to do with translocations per se.
Yeah, okay, that makes sense. So I know Dr. Bergshagel talked about MYC, and being able to find MYC was only because there were more detailed tests available, and before you couldn't even test for it. So are you finding new things with the deeper level of testing that's available now?
Well, let me just tell you the MYC thing real quickly. I think I told you that he and I talked about this a lot, and we decided that MYC was really important, and that we should look at it. And at that time, it wasn't known that there were rearrangements of MYC. It was very hard. There weren't good methods for looking at rearrangements, and so we decided to look for mutations in MYC, and we found some mutations, but they turned out to be germline polymorphisms, so normal alleles. And that was disappointing. But we decided that, well, since we have these mutations and we can distinguish the 2 alleles, let's look at that. And what we found in our cell lines was that when we found mutations that distinguished the maternal and paternal MYC alleles, they only expressed 1, and in almost all cases. So then we decided to develop fish assays to look for MYC rearrangements, and they turned out to be very complicated, and they had not been picked up for the most part before. And so we then found them there, and then another group came in and looked at AMGUS, and they saw very few in AMGUS. So the idea was that these were secondary events. We also saw heterogeneity in myeloma tumors. And then when Leaf developed the mouse model, it turned out that it wasn't thought that was going to create myeloma. It was an MGUS-prone strain, and it thought it was going to create a different disease, but it turned out it created myeloma. So upregulating the MYC created that, and then when you looked again at the gene expression profiling, MYC was up higher. So then recently what we took advantage of is a combination of CGH initially, some comparative genomic hybridization that looks at copy number changes. And we saw it was amazing because about a third to 40% of myeloma tumors, full-blown myeloma tumors, and I think that's important, not necessarily the earliest stages of myeloma tumor. I'm not talking about MGUS now, had copy number changes in the MYC region, and they were pretty scattered. And then in addition, there were FISH analyses for rearrangements involving IgH and immunoglobulin heavy chain and light chain loci that showed that there were even more. So somewhere in the range of 40, probably 50% of myeloma tumors where you can get enough sample to study have MYC rearrangements. Mm-hmm. And so we now think that MYC is the most common abnormality in myeloma. We think that it is that MYC upregulation, which can be biallelic, doesn't require a rearrangement, but the rearrangements happen, and we know some of them are happening during progression, but we know also that some of them are, it's very likely, but this remains to be proven, are causing that transition from MGUS to myeloma. That's our theory anyway. And so there are studies going on now that the MMRF has sponsored to look at whole genome sequencing, and the preliminary results there is they're finding these MYC rearrangements there also now. And so and it's a little bit discrepant with previous studies that were done in France using FISH analyses. They found lower levels, and I think the theory now, for me anyway, is that what MYC is doing is it's not causing increased proliferation. That's very clear. There's no correlation of MYC expression and proliferation. And instead, what I think it's doing is it's increasing autonomy of the tumor. And it's enabling the tumor to go from a small tumor to a larger tumor outside of beyond the normal bone marrow niche. It's still in the bone marrow, but now instead of being a few percent of the cells in the bone marrow, it's now 10, 20, 30, 40, 50%. And I think a MYC rearrangement is giving it a degree of autonomy. And the reason that I think that the French group saw a lower incidence is they were looking at probably their samples are skewed towards an earlier stage of the disease. I mean, that's a theory. It's a way of... But anyway, the bottom line is that MYC is obviously a key, I think, in the pathogenesis of myeloma, and distinguishes it from MGUS in that sense. And that it's hard to target, but there are some promising there's promising agents now that suggest that you might be able to target MYC, which and I think that's gradually happening. We'll see if it pans out.
Well, I guess that would be, we have a few caller questions that I know people are going to want to ask at the end, so I want to leave some time for that. But I guess my question would be for you, with this information about MYC and all this detail, and this is such deep detail about myeloma, I'm just so impressed with it, to be able to subdivide it like that and understand all the pathways that it's doing. Where do you go from here?
Well, here's what I think. I mean, and I think knowledge is a great thing. I think the more you know about things, the better chance that you are to be able to do things. I'm a deep believer in basic science and trying to understand how things work. What is there, because I don't think you can predict where the next lead is going to come from, and I think that doing those basic studies it gives you the chance. That being said, I think that the amazing thing that's happened in the last 10 years is the development of drugs like the lenalidomide, thalidomide, Velcade, and so on. They're targeting the plasma cell. They're not targeting a specific kind of myeloma generally, although they might be more active in one type of myeloma than another, but they're targeting what I would call the plasma cellness of the tumor. So I think that those kinds of therapies are, if we could understand them better, are really important. I think another target is the interaction of the myeloma cell with the bone marrow environment, which is pretty much of a mystery. We really don't have a firm understanding of that. I think that's really important. And I think the third place that's really important is immune therapies. I think that the day of immune therapies has arrived, and there are definitely tumors where immune therapy is very effective, and there's a good reason to believe that immune therapies might be useful in myeloma. I mean, I think it's important to remember myeloma cells are plasma cells, and if you could wipe out all of the plasma cells in your body, including the myeloma cells, that wouldn't be a disaster. You can live without plasma cells. You can do antibody injections and so on. I mean, not that it's ideal. And that's better than if you had a lung tumor. You can't knock out the lung. I think that there are many options for therapies. I think targeted therapies is a great idea, but I think single targeted therapies just generally haven't worked, and I think there's a lot of work going on trying to identify ways of targeting multiple targets at the same time. And so, that's kind of my view of this. But, the future is so hard to predict, and I never would bet against the science and medicine coming up with new therapies. And I think it's just hard to predict when they're going to be there, but I think they will be.
Well, and that's, I guess, my recommendation for patients is if you are educated and you understand the biology of your own disease, then when something new comes out, you can say, oh, well, I sort of qualify for that. and that might be appropriate for me because I have this particular translocation, or I know that, I'm hyperdiploid or whatever.
That's absolutely true. And I think it's like patients can actually contribute to the treatment of their disease, because when they ask questions, they can stimulate people to think, and they have a great vested interest, more so than anyone else, in their disease. And I'm very serious about this. And there are examples of patients who've come up with ideas that researchers and clinicians have used that have been beneficial. So I think that patients need to understand their disease, and they can definitely make a contribution to improving the treatment of the disease.
Well, I think that's the whole theory behind this this program, is that patients can be major contributors, because researchers are brilliant and they're amazing and they're doing this wonderful work. But unless we participate by helping them speed things along, it could take 20 years to do something that could have been done in 10. Or
we all have
5
blind
or 2.
spots. We all have blind spots. The story about the elephant and the people feeling the different parts of it. Well, we all have a perspective, and the patients have an important perspective. There's no doubt.
Well, thank you. I would like to open it up for caller questions.
Sure.
So we have a caller at 983-6757, and if you have a question for Dr. Kuehl, call 347-637-2631 and press 1 on your keypad. Okay, so our first caller question. Go ahead.
Hi, doctor. This is Dana. How are you? Hi, Jenny. I have a question.
Sure.
Is there any other? I was very interested in what you were discussing about cyclin D1. I'm smoldering, so I'm really just trying to learn as much as I can. And in my situation, I'm wondering if there's any other process that could result in the upregulation of cyclin D1 other than the primary translocation 1114. I have cyclin D1 on my immunohistochemistry. They found up to 15% total plasma cells on the core sample, 6% in the aspirate sample. And this is a 2-year-old bone marrow biopsy. But I had a normal fish. So in theory, from what I understand, we should have found the translocation 1114 in my fish. I reached out to a pathologist who ran a plasma cell labeling index for me as a courtesy on this actual sample, and the results were 0%. So he explained to me that I had a very low proliferation rate, but stated that my cells were expressing cyclin D1 like crazy, quote, and that was, what he basically explained to me. And I needed the layman's terms, of course. Yet 97% of my cells were ID'd as aberrant on the 8-color flow. So I guess depending upon these risk progression models, since I am in the smoldering phase, that's what I kind of try to focus on. The flow indicated that I had a higher risk of proliferating and progressing. So at this point, I'm 2 years into this, 2 years and a couple of months, and my M spike and my abnormal free light chain ratio have been relatively stable. And I'm really just trying to kind of grasp anything. Like at this point, what do I need to know about my biology in order to kind of move forward and be smart about this?
Well, okay, so let's take your first question, which is about the cyclin D. So hyperdiploid tumors can make cyclin D. Now, usually it's lower than if you have a translocation, and so there's a clear-cut difference, and there's a cutoff, and it can go to quite low levels, but it can be moderately high. So it's possible. I don't know if they know whether your tumor is hyperdiploid or not, but the fact that they didn't detect a translocation could mean that you have a hyperdiploid tumor that happens to be making a relatively high amount of cyclin D. Another possibility is that when they do the test, this would be very rare, but the test they do for cyclin D probably is looking for IgH cyclin D1 translocations. But rarely it's possible that you could have an IgL translocation. That is a light chain or lambda or kappa, well, it's very rare. And not only that, but the other thing that I should say is that it is my belief that that translocations that are so-called primary translocations, sometimes that the genes that are involved in primary translocations can also come up in secondary translocations. That is, they didn't come up as primary, they came up as secondary, and it's my belief, and this is based on our studies on MYC, where they're not all IG, many of them are not IG, but they involve a limited number of other loci. There's no test for those now. So it's possible that you would have a cyclin D, that they've missed the translocation. Okay, it may not be IG or whatever. Now, that being said, just with respect to your disease, I mean, I think that it sounds like you're getting the flow cytometric test that stratifies patients, and they're telling you that you're in the group that's more likely to progress.
Exactly. Yes.
Well, what I would say is that what you want to do is you want to make sure that you get careful follow-up. There are trials out there now where they're starting to do trials on patients that have smoldering who are in this high-risk group, and they are starting to include some of those patients on trials. And, when you enter a trial, that is the patient's choice. In other words, so you want to apprise yourself of what those trials are and what the status of them is. Find out from the people who are doing these tests on you, do they recommend that you would do that trial? And then there are a variety of places that are doing these trials now. Some at the Mayo Clinic, I think. I think some here at the NIH. Ola Landgren has done some. He's moving to Sloan Kettering, so
yes.
I know he's doing some of that. So I think just make sure you get the best possible medical care, that you get good follow-up, and then make sure you read about these things, and so that you can ask intelligent questions of your doctor. That's really important.
Now, what about additional cytogenetic testing? Do I need to pursue that? Should I have another bone marrow biopsy? Do I need gene expression profiling to kind of bring better clarity to my situation?
Well, I mean, I'm biased in this regard. I mean, I believe more information is better. And so my feeling is more information can't hurt. well, I mean, apart from having your bone marrow taken, but
yeah.
but in all seriousness, I think the more you know, the better, and because I think that is important. Now, that being said, there isn't necessarily it isn't necessary that at this point in time that it's going to make a lot of difference, because maybe there aren't treatments right now, but they may become, things may change. And so I think having that information, I don't think it's a big deal, and if you can get it done and, and your insurance and whatnot pays for it, I would do it.
Okay.
That's what I would do if I were in your situation.
I thank you for that. Thank you so very much.
Okay.
Okay, thanks for your question. Okay, our second caller. Go ahead with your question.
Oh, hi. So that was an awesome answer, and it kind of feeds into the question that I have. So because the question was really, what do I do differently as a patient if I know my disease biology, and that was like a very good case in point about what I might know and what I might do. Could you, because I've listened to the shows and you've talked a lot about the disease biology, but that was like the first example of what I might do differently now that I know it. Can you give us other examples about, the importance of understanding disease biology and how that really affects the treatment path?
Well, I mean, I think, I think that preliminarily, myeloma is divided into high risk and low risk. So high risk would be, and this is based a lot on cytogenetics, because those are the tests that have been available. And so high risk is 414 translocation, it's MAF translocations, it's P53 mutations. And sometimes chromosome 13 abnormality, although that's thought to be mainly because that's associated with MAF and FGFR3. And then you have the low risk. The low risk are hyperdiploid and cyclin D1. Now, usually it's not routine to do studies for hyperdiploidy, and so mostly people have done the studies for translocations. They've done P53 to look for loss of allele of P53, which is associated with progression of the disease. Chromosome 13 loss is done, although that isn't as important if you do the 414 and MAF translocations. But I think that's important to know. Again, what I would say, though, it depends who you're working with. I mean, I think I think as a patient, you want to know what's going on. You want to push your doctors. You want to ask them questions, and you could ask me, and I'd say, if I were, people come and ask me periodically. They'll call me. They've read something I wrote, and they'll say, "Can you give me some advice?" And one of the things I would say is, I don't treat patients, but I have a philosophy, and I also know where I would go if I had myeloma. And I think what I would say is, you want to get the best possible treatment. You want to know as much about the disease as possible. I think it's important to have as much information so that when more information can never hurt, and obviously there are limits on cost and et cetera, but anyway, I think what I said to the previous patient is I would say the same thing to you. I mean, I think that learn about the disease, talk to people, and ask questions of your doctor, and if you're not satisfied, get another opinion. Don't be afraid ever to get another opinion. Any good doctor would never discourage you from getting a second opinion. I don't know if that's helpful, but anyway, that's...
Yeah, no, it's a great answer. And I think, Dana and Jenny may be the most informed patients on the planet. So I applaud what you guys are doing, and keep up the great work.
Well, yeah, I mean, I think, like, follow the MMRF. They're great. I think that they've just made such an incredible impact. Kathy Justy is out of sight as far as I'm concerned. So I think there's a lot to learn. I mean, I'm not putting down any other organization, but I'm very impressed with what they've done. And I mean, it's focused on research, and which I think is really key.
Yeah, that's great. Well, thank you.
Okay.
Okay, and we had one, I know we're over time, so I'll ask this quickly, but we had one write-in email question that said, has the NCI ever considered putting together patient data similar to how the MMRF Compass study has done, but on a larger scale?
Not that I know of. I don't know that that's necessary. I think what the MMRF has done, I don't think the NCI is going to do better than they've done. I mean, I think what they've done is spectacular. And
I've heard that.
I think that the whole landscape of myeloma research has just changed since they got involved. I still remember years ago that they assembled all the myeloma grants at NIH, and I thought one of the amazing things was there were very few, and there were very few actually on myeloma itself. But one of the things is it turned out when I looked at the list, there were numbers of melanoma in it, which is is a skin tumor.
Oh no!
So I said to someone, I said we've really made it in myeloma when people are no longer confused about melanoma and myeloma. So anyway, I don't think the NCI is. I think the MMRF is doing the job.
Well, I've heard from several researchers that they love their researcher gateway and all the data that comes from their compiled data. So I applaud what they're doing. It's really amazing work.
Yeah, and there's just no doubt about it.
Well, Dr. Kuehl, thank you so much for joining us today. We really appreciate you taking the time to help us understand the biology, why it's important to us, and then what we do with that information. So thank you for helping us identify the fact that all myeloma is not the same, so we can get to more personalized care.
Well, it's a pleasure, and I've always said that I get a great deal of satisfaction when a patient calls me and asks my opinion, and I always tell them, well, my opinion probably is not worth much more than I charge for it, which is nothing. But I hope that's not strictly true. But I'm really glad to be able to do this, and if it's helpful, it's great.
It's very helpful to patients. So we really appreciate you taking the time.
Okay.
Well, thank you for listening to another episode of Innovation in Myeloma. Join us next week for our next mPatient interview as we learn about how we as patients can help drive to a cure for myeloma by joining clinical trials.
Lucky Land Casino, asking people, what's the weirdest place you've gotten lucky?
Lucky? In line at the deli, I guess?
Aha, in my dentist's office. More than once, actually.
Do I have to say?
Yes, you do.
In the car before my kids' PTA meeting.
Really?
Yes.
Excuse me, what's the weirdest place you've gotten lucky?
I never win and tell.
Well, there you have it. You can get lucky anywhere, playing at LuckyLandSlots.com. Play for free right now. Are you feeling lucky? No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.